Okay, welcome everyone. My name is Frank Gonzalez from the National Cancer Institute. I'll be introducing today's speaker, Dr. Jeff Peters. First, I want to remind everyone that after the talk, there's going to be a reception back there that's going to have cookies and drinks and so forth. I encourage everyone to attend and have an opportunity to speak with Dr. Peters. Jeff is a California born and bred. He got his BS degree and his PhD in nutrition from the University of California at Davis. He then came to the NCI after a short postdoctoral stay with Lynn Willie in Davis, where he studied for 4 years. He then moved to the Penn State University, where he rose to the ranks of full professor. Jeff has received a number of awards for his work, including the Society of Toxicology Achievement Award. He's on NIH study sections. He's editor on the editorial boards of several journals and is associate editor of Toxicological Sciences, a very well-respected toxicology journal. I'd just like to take a moment for all the fellows out there to note that Jeff was in the NCI as a postdoc for 4 years, and he published more than 30 papers, which kind of sets a benchmark for people in my lab. Now, many people will argue that it's a quality rather than quantity that matters, but in fact, many of Jeff's papers have been heavily cited in the literature, and he's continued this trend of high productivity with a large number of papers since he's joined the Penn State University, and he's well funded by the NIH. Today, Jeff will talk about this fascinating group of nuclear receptors, of family receptors called the PPARs, that are not only of immense academic interest, with thousands of papers published on these 3 proteins, but they're of great industrial interest because they're the targets for drugs that are on the market for treating metabolic disorders like diabetes and hyperlipidemia, hypercholesterolemia, where they pull in billions of dollars in sales. So these are a very interesting group of proteins, and I'm hoping Jeff will further convince you of this during the next 40 minutes or so. So Jeff?
Thanks. Am I on here? You can all hear me? Okay. Well, thanks for that kind introduction, Frank. I have to tell you that standing up here to calm my nerves for a couple seconds, it kind of takes me back to the days when I was actually sitting out in the audience listening to speakers up here. It kind of reminds me, it wasn't that long ago, 4 kids later, I guess, and then I'm now up here. It's a great opportunity to be able to come and speak to this group. And rather than speaking today about specific research projects that are undergoing in my laboratory, I thought I would take the time to summarize the field of PPARs and hopes of convincing you, or at least enlightening you about the potential of targeting peroxisome proliferator activated receptors for the treatment and prevention of diseases. So my presentation will begin with a brief background and history of the PPARs, and I will follow that up with several sections summarizing the functional roles for PPAR alpha, PPAR gamma, and PPAR beta, which is also referred to as PPAR delta in the literature, but I will refer to it as PPAR beta for the duration of my presentation. And I will describe the functional roles for these receptors in metabolism and cancer, and I'll follow that up with a short section at the end discussing the potential application of dual or pan PPAR agonists, and then the future of PPARs. So the history of PPARs really begins in the early 60s when a group in Switzerland noted that administration of a hypolipidemic drug caused hepatomegaly, and more importantly, they noted that in the liver of these animals there was a significant increase in the number and size of peroxisomes. They described these as microbodies at the time. And they didn't really know what that meant, and they probably had no idea that they were going to be leading the field and leading us down this road, but that was the description that got this thing started, more or less. And about 10 years later, Jan Reddy, one of the pioneers in this field, made the observation that in addition to that compound that group noted, there were actually a number of structurally unrelated compounds that were capable of producing the same type of effect in the liver of these animals, and that they saw hepatomegaly and peroxisome proliferation. And it was at that time that they coined that phrase, peroxisome proliferation, to describe this phenomenon, and then also to describe these compounds that were capable of causing this effect as peroxisome proliferators. Shortly after that observation, Jan Reddy's group then went on to discover, very importantly, that chronic administration of these chemicals caused not only the peroxisome proliferation, but they noted that these animals that were fed these chemicals for long periods of time, the increased incidence of hepatocellular carcinoma and adenoma in the liver of rats and mice. And now, they didn't know the mechanism underlying this effect at the time, but they hypothesized that this was a receptor-mediated event. And the reason for that, or the rationale for that idea, was based on other findings at the time where the thyroid hormone receptor, for example, had recently been identified. And the idea was that xenobiotics were functioning through some type of receptor. And so at that point, in that time, they said, well, they started to try to find this receptor, and the race began at that point to try to identify the receptor that mediated this event. And he was right. They were right. There was a receptor that caused this effect. And that was found about 10 years later by a group in England who, using the estrogen receptor DNA binding domain as a tool, they fished out this receptor from a cDNA library from rat liver treated with these peroxisome proliferators. And indeed, they found that this receptor was capable of transactivating in response to the chemicals that these investigators had noted caused the peroxisome proliferation and hepatomegaly. They got an increase in activity that was associated with that, and hence they coined the phrase, or they termed this receptor peroxisome proliferator activator receptor. It wasn't coined alpha until later, but at that time that's where they were at. So it turns out that PPARs belong to a class. There are a class of them, and there are actually 3 of the PPARs. And so human PPAR alpha, there's PPAR beta, and there is also PPAR gamma. They share significant homology in their DNA binding domain, similar to other nuclear receptors. And where they are a little bit more divergent is in their ligand binding domain. And it was actually this region of the genomic DNA that we took advantage of when I was here during my days at the NIH to make targeting vectors, to make knockout mice, to study the functional roles of these receptors. In addition to the ligand binding domain feature of this particular part of the protein, though, there's also another important part down here at this business end, which is important for interactions with co-repressors and co-activators, in addition to its ability to heterodimerize with RXR alpha, its partner. And that is shown very nicely on this slide from some recent work published in Nature showing the crystal structure of the PPAR with its heterodimerization partner RXR alpha on a peroxisome proliferator response element on DNA. And as you can see on this slide, the PPAR is depicted in red and the RXR alpha is depicted in blue. There are a couple of points to make from this. And one of those is that you'll note that the ligand binding domain is in relatively close juxtaposition to the DNA binding domain on the PPAR, but as compared to the RXR alpha, the ligand binding domain is a little bit further away than the DNA binding domain. And the second point to make from this slide is that, in fact, the ligand binding domain is the region that is important for heterodimerization with RXR alpha, and that was really known from deletion analysis, but it was confirmed by this nice paper describing the structure of PPAR. And as I mentioned earlier, PPARs belong to a class of nuclear receptor family that range from the androgen receptor, estrogen receptor, retinoid receptors, FXR, LXR, vitamin D receptor, and thyroid hormone receptor, to name a few, and PPARs belong to the same class of nuclear or soluble nuclear receptors. What differentiates them, or one of the things I should say that's actually similar, is they all contain, as I noted before, they all have ligand binding domains, DNA binding domains, and transactivation function domains at either end. But one of the main features that separates them all from one another is their ability to be activated by specific agonists. So for the case of androgen receptor, there are androgens. For FXR, it's bile acids. vitamin D receptor, it's vitamin D. And in the case of PPARs, it is thought that fatty acids or fatty acid derivatives are the endogenous agonists for these receptors. And this slide illustrates the mechanism of transcriptional upregulation of target genes by PPARs. PPARs are thought to exist in the cytoplasm or the nucleus in association with co-repressors. And in response to ligand activation, the receptor undergoes a conformational change and dissociation of co-repressors. This leads to heterodimerization with its another nuclear receptor, RXR alpha, and this heterodimered ligand-bound receptor complex will then recruit coactivators that contain histone acetyltransferase activities. They will bind to peroxisome proliferator response elements on target genes. This leads to the recruitment of RNAs, or I should say acetylation of histones, which leads to the remodeling of chromatin and loosening of the chromatin structure. Then RNase polymerase comes along, and you have translation of, or transcription of mRNAs, and translation into proteins, and ultimately you end up with biological functions. And this particular mechanism of action holds true not only for PPARs, but also for many other nuclear hormone receptors. In some cases, it is through homodimerization. In the case of PPARs, it is in heterodimerization. In addition to that direct transcriptional upregulation, though, PPARs are also known to epigenetically interact with other proteins. So in the case, on this example here, TNF-alpha is known to activate the NF-kappa B transcription factor pathway, and in doing so leads to expression of pro-inflammatory signaling molecules. PPARs interact with this pathway by the fact that the ligand-bound PPAR can physically interact with the NF-kappa B subunit of the P65 subunit of NF-kappa B, and in doing so attenuate NF-kappa B dependent signaling, and through this mechanism functions as an anti-inflammatory type thing. Now, it should be noted that similar pathways have also been described for other pathways such as AP1, where PPARs also interact with. In addition, there are other pathways that are out there that have described potential mechanisms of anti-inflammatory activities, but I don't have time to go into great detail for today's lecture. Now, there is considerable redundancy in the response elements of PPARs, which becomes important a little bit later in my talk. And as noted from this slide, this is the consensus sequence for transcriptional for target genes of PPARs. Now, if you overexpress a PPAR in any particular cell type and present that cell with ligand, you can turn on target genes that are presumably specific for one PPAR. That has led to the idea that the cellular expression of different PPARs and the relative abundance of the endogenous agonists are the two levels, or at least two levels of regulation for PPAR specificity in target tissues. For example, in the liver, PPAR alpha is predominantly at highest levels in the hepatocyte, and PPAR alpha has a significant role in regulating pathways in the liver. Whereas in the intestine, PPAR beta is the predominant isoform, and here, PPAR beta has an important functional role. And then in adipose tissue, PPAR gamma is expressed at the highest level, and similarly, PPAR gamma has an important role in regulating adipogenesis, as I will describe a little bit later. Now, I mentioned earlier that fatty acids are potentially ligands for PPARs, and this is based on some work done in Ron Evans' laboratory by Barry Foreman, who showed that using a simple system of hooking a reporter assay or response element for PPARs to a luciferase construct and co-transvecting this into cells in the presence of different agonists, they were able to actually examine what happened in response to fatty acids. They could measure this by activity. And what they found was that a number of different fatty acids, and I'm just showing you a subset of these from their work, that indeed when these cells are presented with different fatty acids, activity of PPAR alpha is increased, in contrast to that with PPAR gamma, where only arachidonic acid was able to induce a small increase, and similarly with PPAR beta, they got a similar profile. So fatty acids can indeed activate this receptor, and this has been confirmed subsequently by others. Now, what's important to note is that the concentration required for this effect is in the low micromolar range. And the reason I bring that up is because more recently, in recent years, they have developed more high-affinity agonists for PPARs. So I noted earlier that the hypolipidemic drugs that were actually used to identify the receptor that led to the discovery of the receptor were the fibrate class of hypolipidemic drugs, which are shown here, and they activate PPAR alpha in the micromolar range. This compound down here, GW7647, is a very high affinity agonist for PPAR alpha, and it actually has preferential affinity for human PPAR alpha as compared to the mouse. The L compound here, 165041, was the first high-affinity PPAR beta agonist described in the literature with affinity in the nanomolar range. And GW501516 and 0742 are also capable of activating PPAR beta in the nanomolar range. And then for PPAR gamma, troglitazone was one of the first high-affinity agonists described for PPAR gamma in the literature that was actually used clinically, and it was withdrawn from the market due to idiosyncratic liver toxicity that I'll talk a little bit about later. So that's troglitazone, as compared to pioglitazone and rosiglitazone, which are currently on the market and used for the treatment of type 2 diabetes. So these compounds here are capable of activating PPAR gamma in the low micromolar, high nanomolar range. Excuse me, I'm getting over a cold, so actually I'm still pretty heavy with one, so I'm a little bit hoarse here. So PPAR, I'll begin my section talking about PPARs here. Now, PPAR alpha is indeed the molecular target for the fibrate class of hypolipidemic drugs. And what happens in response to ligand activation, transcription of fatty acid catabolizing enzymes is increased, and this leads to the degradation of fatty acids, and this is known to occur in the peroxisome. And in doing so, the byproduct of acyl-CoA oxidase, one of the key enzymes in this pathway, is the generation of hydrogen peroxide, which is important for a little bit later in my talk. But in addition to regulating peroxisomal beta oxidation, ligand activation of PPAR alpha is also very important in regulating mitochondrial beta oxidation. So it encodes for, transcriptionally upregulates a number of similar genes that are encoded for in the peroxisome in the mitochondria that catalyze very similar reactions, but in doing so, what separates them from the peroxisome is that you generate high-energy phosphate. And this is important in generating energy, in particular during periods of starvation, when your body needs that, when certain cells need to generate energy. Oops, let's go back to that one. So PPAR alpha is very important in regulating serum lipid concentrations, and in fact, that was the basis for the use of the fibrate class of hypolipidemic drugs. So this is just shown here in this slide, is that in response to ligand activation, you get a significant decrease in serum triglycerides, and that in the absence of PPAR alpha expression or in the knockout mice, this decrease does not occur. That demonstrates the essential nature of this receptor in this response. The second take-home point from this slide is that constitutive levels of triglycerides are significantly higher in the knockout mice. That suggests that PPAR alpha also has a functional role constitutively in regulating serum lipids. And that was confirmed later by some other papers we published showing that it actually regulates those same proteins involved in mitochondrial fatty acid oxidation, and in the absence of the receptor, that accounts for this increase in serum lipid levels. So PPAR alpha is essential for fatty acid catabolism. In the adipose, it regulates fatty acid transporters and fatty acid binding proteins, allowing for, during periods of starvation, for fat triglycerides to be transported to different tissues, such as the heart, skeletal muscle, or particularly in the liver, where PPAR alpha also regulates the same proteins in addition to the mitochondrial fatty acid beta oxidizing enzymes and the peroxisomal enzymes that I mentioned earlier, as well as apolipoproteins. So PPAR alpha is very important in regulating this constitutively in the body, and that occurs on a regular basis every day. Now, we noted in about 2000 we discovered a mutation, a polymorphism in PPAR alpha that we thought was going to be a conservative mutation because it encoded for a simple leucine to valine substitution in one amino acid in the DNA binding domain. However, when we transvected this construct into cells, what we found was, as compared to the wild-type cells that responded with normal increase in activity, this particular construct led to increased transcriptional activity, which was a bit of a puzzle. And this observation was actually repeated by 2 separate labs in addition to identifying the same polymorphism. So we know it's a real event. Now, characterization of this mutation has been ongoing in the last 10 years, and there are a number of papers out there describing potential interactions. In particular, associations with the efficacy of fibrates, obesity, and serum lipids. But the majority of these papers out there are more or less equivocal in the sense that some papers find positive or negative associations, but the findings are relatively inconsistent. More recently, it's found, or that studies are doing, is they're examining the relationship of this polymorphism with other proteins involved in fatty acid metabolism, such as fatty acid binding protein, and they're finding that it's more likely to explain that there are going to be more than one gene involved that explains these interactions. And this is an area of active research. So in addition to regulating serum lipid, or I should say lipid homeostasis, PPAR alpha is also essential in mediating peroxisome proliferator-induced hepatocarcinogenesis. And this is from some work that we did here at the NIH about 10 years ago in showing that in wild-type mice administered these chemicals, you see basically 100% incidence of hepatocellular carcinoma or hepatocellular adenoma in these mice. That's been known for years. But what was very interesting was when you fed these chemicals for the same period of time to mice that lack expression of this receptor, essentially no tumors are found in these mice. And this observation is fairly profound, and it demonstrates the essential nature of PPAR alpha in mediating the liver cancer associated with these chemicals. So the first thing that comes to mind is, well, how can that be used? And if these chemicals are still being used today in the market, how can that be? Is it a cancer risk? And it turns out that there has been an enormous amount of research undergone in the last 10, 20 years that has looked at this. And the evidence to date suggests that humans are likely refractory to peroxisome proliferator-induced hepatocarcinogenesis. That's based on a number of studies, including epidemiological and prospective studies in humans showing a lack of relationship between fibrate administration and liver tumorigenesis, as well as in vitro and in vivo models showing significant species differences between rodents and humans, as well as differences in expression levels. It's thought that humans might express substantially lower levels of PPAR alpha in the liver as compared to rodents. Now, some of these observations are somewhat equivocal, but more recently, some recent work that was done, I'll talk about in the next slide, suggests, using the PPAR alpha humanized mouse model, suggests that there might be a mechanism to explain this difference. So in rodents, administration or ligand activation of PPAR alpha causes an increase in intracellular levels of hydrogen peroxide due to the increase in acyl-CoA oxidase in the peroxisomes. Now, whether or not this leads to oxidative stress and oxidative DNA damage is something uncertain to date, but it remains possible that something that may contribute to DNA damage. What is known, and what's been known for years, is that administration of PPAR alpha agonists causes an increase in cell cycle progression. That's been known because it's a causative factor in liver tumorigenesis observed in these animals. What was worked out recently by Dr. Yatrick Shah in Frank's lab was the mechanism involved in this. And he showed that PPAR alpha activation led to a decrease in the LET7C microRNA cluster, which led to an upregulation of proteins such as MYC, which promotes cell cycle progression. And when combined with either the DNA damage that occurs in response to oxidative stress or endogenous DNA damage that is not repaired, combined with this increase in cell cycle progression, causes liver tumor formation, such that PPAR alpha in the rodents or mouse modulates lipid metabolism as well as an increase in cell proliferation. In contrast, using mice that express a human PPAR alpha but not the mouse PPAR alpha, Dr. Shah demonstrated that this decrease in LET7C microRNA does not occur, this increase in cell cycle progression does not occur, and most importantly, the humanized PPAR alpha mice do not exhibit liver tumors after years of feeding these chemicals. So what that suggests is that the human PPAR alpha modulates lipid metabolism but not cell proliferation. Mechanistically, it must have something to do with the receptor. They're not quite sure exactly what that might be at this point, and importantly, I think that these findings need to be examined in other human models, but clearly these results have shed significant light on our understanding of the potential mechanism underlying the species difference between rodents and humans. So for PPAR alpha, it's a very important protein involved in regulating fatty acid catabolism that regulates serum lipid levels. It's involved in actually increasing HDL cholesterol via increases in expression of apolipoprotein A1, It has anti-inflammatory activities, which I can't really describe today's lecture due to time constraints, and it is involved in rodent-specific liver carcinogenesis. So moving on to the next PPR, that would be PPR gamma. As probably know a lot more about this one as well. This was in the early 1990s, shortly after the cloning of PPR alpha, from some work done by Peter Tontono's in Bruce Spiegelman's lab up at Harvard. He showed that if you took fibroblasts and overexpressed PPR gamma, that you could cause these cells to differentiate into adipocytes. And that's shown by this slide here, where there's an increase in oil red O staining. If he did that with RXR alpha or PPR alpha, he did not see the same effect. So this was the first evidence to demonstrate that PPR gamma promotes terminal differentiation of adipocytes. And it turns out that PPR gamma is actually the molecular target for the thiazolidine diones. Now, thiazolidine diones were discovered by screening compounds that were capable of producing hypoglycemia in a diabetic model, but it wasn't until years later that they discovered and made the connection that they were actually, the thiazolidine diones were ligands for PPR gamma. And the question became, shortly after this discovery, was, well, how does PPR gamma mediate its hypoglycemic effect? Now, what is known, and there's actually a gray area with that today, but I'll shed light on that in a little bit here. What is known is that in response to ligand activation of PPAR gamma, there is an increase in expression of a number of target genes in the adipose, including lipoprotein lipase, fatty acid transport protein, and CD36. And that leads to an increase or an influx of fatty acids into the adipocytes, causing the differentiation of adipocytes, so you end up getting more smaller adipocytes. Now, if you delete expression of PPR gamma in the adipose, you diminish the hypoglycemic effect. And what that demonstrates is that, or suggests is that the adipose is the primary target tissue of PPR gamma. So the generic, how does that really work? Well, again, this is a bit of a gray area, but what is known is that the generic diabetic phenotype involves a decreased insulin sensitivity in adipose, a decrease in glucose uptake, and an increase in the release of fatty acid, which is reflected in the serum by increases in these constitutives, including serum fatty acids and the serum adiponectin, as well as TNF-alpha, which is increased. In the liver, there is a decrease in insulin sensitivity and an increase in glucose output, and in the skeletal muscle, there is a decrease in insulin sensitivity and an increase in, or a decrease in glucose uptake, which confounds factors and contributes to this increase in serum glucose levels. What happens in response to ligand activation is that there is this net influx of free fatty acids into the adipocytes, and there is an increase in the synthesis of adiponectin, which may or may not be a direct target gene of PPAR gamma. In addition, there is a decrease in TNF-alpha production in the adipocytes that may be due to actually macrophages present in the adipocytes, and that has reflected all these changes by causing a decrease in serum fatty acids, this increase in serum adiponectin, and a decrease in TNF-alpha, and that leads to an increase in insulin sensitivity in the liver as well as in the skeletal muscle. Now, the direct target genes involved in this, again, are not known, but it is thought that adiponectin might be one of the key players involved in this. So while PPR gamma has its great associations with improving type 2 diabetes, it's also important to note that there are significant adverse side effects associated with thiazolidine diones. So as I noted earlier, hepatotoxicity was associated with Rezulin or triglydzone and was the basis for its withdrawal from the market. In addition, though, in a small cohort of individuals that are being treated with these chemicals, it is noted that there can be significant fluid retention that may be due to an increase in epithelial sodium channel pump in the kidney. There can be edema, which is confounded by fluid retention, as well as changes in endothelial permeability. Weight gain, which might be due to the increases in adipogenesis as well as fluid retention. And all three of these factors contribute to a higher rate of congestive heart failure in patients being treated with these chemicals. And lastly, more recently in the last few years, it's been noted that bone fractures due to potential increases in osteoclast activity have been observed in populations treated with these chemicals. So there are significant adverse side effects associated with thiazolidine diones, and they must be closely monitored in the clinic. And as with PPR alpha, there are similar roles described in the literature for PPR gamma in cancer. Now, initially, it was thought that activating PPAR gamma caused or promoted tumorigenesis. And that was based on some papers published in the late 1990s suggesting that activating PPAR gamma caused an increase in colon tumors in the APC-min mouse model, as well as the suggestion that activating PPAR gamma by eicosanoids promoted tumorigenesis. However, this started a flurry of experimentation by many, many different laboratories. And what was found subsequently is that, in fact, activating PPAR gamma prevented tumorigenesis in these models, as well as if you delete PPAR gamma expression in the intestine, you get enhanced tumorigenesis in these models. In addition to many other studies performed by other laboratories demonstrating other types of chemopreventive effects, has led to the consensus that PPAR gamma actually attenuates tumorigenesis. And the mechanisms underlying this are primarily mediated through a couple of different pathways, in that PPAR gamma agonists can cause terminal differentiation, they can increase apoptotic signaling, and they can inhibit cell proliferation. However, the direct target genes that mediate the inhibitory effects of PPAR gamma agonists are still really unknown. What is known is that there are also some independent events, and so using siRNA approaches or knockout mouse models, people have actually established that PPAR gamma agonists can also exhibit or can cause chemopreventive events independent of PPAR gamma. And these primarily are focused on inducing the induction of apoptosis as well as inhibition of cell proliferation. So there are really two potential pathways where these things are affected, but they have led to a number of different studies examining the efficacy of PPAR gamma agonists in human clinical trials. And with that said, the monotherapies, the studies that have examined monotherapies of PPAR gamma agonists have demonstrated modest to no efficacy to date. So this has been examined in a limited number of trials, colon cancer, lung cancer, for example, and the efficacy observed in these studies has been fairly modest. But however, more recently, people have come to the idea that combining PPAR gamma agonism with other chemopreventive or chemotherapeutic targets might lead to greater efficacy of inhibition or chemoprevention, and these studies are ongoing, and that is an active area of research as well. So for PPAR gamma, activating this receptor can improve insulin resistance, and it's thought to be through direct effects in the adipose. There are notable side effects associated with administration of these compounds that must be monitored closely in the clinic. Again, don't have time to talk about it today, but PPAR gamma agonists are also anti-inflammatory, and they are chemopreventive through their ability to induce terminal differentiation, apoptosis, and cell proliferation. And that moves into my last section of my presentation, and that is to talk about the role of PPAR beta. PPAR beta is expressed at high levels in some tissues, and this slide illustrates that. In the small intestine, in the colon, and in the liver, PPAR beta expression is fairly high. And what's interesting to note is that expression of this receptor is found in the nucleus as compared to the cytosol, and it can be co-immunoprecipitated with its heterodimerization partner, RXR alpha, suggesting that constitutive expression of PPAR beta has some functional role in these tissues, and I'll talk a little bit more about that in just a bit. The first functional role for PPAR beta described in the literature was based on some work from Johan Owerk's group, who showed that administration of L165041 caused a significant increase in serum HDL cholesterol through unknown mechanisms at the time. And what was most striking about this observation is that they knew that PPAR alpha agonists could increase HDL cholesterol substantially, but the effect observed with PPAR beta agonists was substantially greater. So this sparked some great interest in the role of this receptor in metabolic diseases. Shortly thereafter, another paper was published showing that ligand activation of PPAR beta promoted reverse cholesterol transport and again increased significantly HDL cholesterol, likely through the induction of ApoA1, and there was a marked decrease in serum triglycerides as well as serum insulin levels. And this led to the idea that targeting this receptor would be a potential benefit for treating metabolic syndrome, because these events were noted in an obese monkey model. Subsequently, a group in Japan showed that ligand activation of PPAR beta led to increases in fatty acid oxidation, specifically in skeletal muscle. And this was an interesting finding. The target genes in skeletal muscle, much similar to those expressed in liver, were found to be regulated specifically by PPAR beta, suggesting that PPAR beta has a functional role in this tissue in regulating fatty acid catabolism. And in their model, they also noted that ligand activation of PPAR beta could decrease diet-induced obesity, it improved glucose tolerance, and it improved insulin sensitivity. And then lastly, more recently, from some work in Ron Evans' laboratory down at the Salk Institute, his group showed that activating PPAR gamma led to a significant increase in the number of oxidative myofibers in skeletal muscle. And this led, when combined with training, led to the ability of these animals to exercise for a greater duration and run for a longer distance. And this is an interesting observation. It raises some ethical issues, potentially to the idea that the use of these compounds could be used for illicit activities to enhance performance, used as performance-enhancing drugs, much as similar to what you've heard about recently in the news. Now, as with PPAR alpha and PPAR gamma, PPAR beta also has potential roles in cancer. One of the first suggestions that PPAR beta could regulate cell growth was provided by the observation showing that the hyperplastic response induced in response to forbol ester in mouse skin is significantly exacerbated in the absence of PPAR beta expression. This suggests that PPAR beta attenuates cell proliferation, and the mechanism underlying this effect is currently not known, but it is likely related to the ability of PPAR beta to induce terminal differentiation in skin. And as shown from this slide, this is probably the most definitive phenotype described to date for PPAR beta. It has been shown to induce terminal differentiation not only in mouse, but also human keratinocytes, but also in intestine and a number of other cell types. So ligand activation of PPAR beta likely inhibits cell proliferation through its ability to induce terminal differentiation. In addition to that activity, as I noted earlier, and this slide could be drawn up for a couple of other PPARs as well, but it's a little bit more diverse for PPAR beta. There are a number of anti-inflammatory activities associated with PPAR beta, including its ability to interact with the P65 subunit of NF-kappa B, but also by some work from a group up at Frederick showing it can inhibit STAT3, BCL6, or ERK5, and combined, all of these activities typically lead to decreases in the expression of pro-inflammatory cytokines, chemokines, and cell adhesion molecules. So, based on the ability of this receptor to modulate terminal differentiation and inflammation, has led to one hypothesis to explain the phenotype or the effect of this receptor in cancer. So one idea out there about the functional role of PPAR beta in cancer is that fatty acids are the endogenous agonists of PPAR beta, and in activating this receptor, it increases expression of proteins such as fatty acid binding protein or ADRP, which are well-characterized PPAR target genes. And these genes are associated with the induction of terminal differentiation, and that leads to attenuation of cell proliferation. And this model is really has been modeled for colon cancer, but a similar one has been proposed for skin as well. In addition to that effect, though, there is the potential for synthetic agonists to activate this receptor and through other unidentified target genes elicit the same response, coupled with the ability of the receptor to antagonize NF-kappa B signaling, for example, leads to inhibition of inflammation. And in doing so, this explains the phenotype reported in the literature showing inhibition of carcinogenesis in response to ligand activation of PPAR beta. And there are a number of reports from reputable labs demonstrating this effect, suggesting that this is the case. In contrast, there is an alternative hypothesis out there in the literature that describes essentially the exact opposite. And that is based on the idea that in response to an APC mutation, which leads the APC protein dysfunctional, there can be an increase in beta-catenin TCF4 signaling, and that leads to an increase in the expression of known oncogenes c-MYC and cyclin D1, which drive progression of cell proliferation and tumor promotion. it was suggested that PPAR beta was a similar target gene of this pathway, and through its activity increased the expression of yet to be identified target genes, and that drives tumor progression. That led to a subsequent hypothesis suggesting that NSAIDs, their ability to inhibit colon tumorigenesis, for example, was due to the fact that they could prevent COX-2 activity, and that would prevent the conversion of arachidonic acid to prostacyclin, which is thought to be an endogenous agonist of PPAR beta. And that explains their inhibition or the ability of NSAIDs to inhibit tumorigenesis. However, since that hypothesis, a number of papers have been reported that are inconsistent with this idea, which I'll touch on a little bit later, and that led to a secondary hypothesis suggesting that NSAIDs actually inhibit PPAR beta expression, and through this inhibition, there is this, again, attenuation of cell proliferation, or I should say attenuation of cell growth that way. So there are two, and again, the latter hypothesis is reported from very reputable labs as well. And so right now, there's a controversy in terms of where or what PPAR beta is doing. And there are a number of major issues out there, and this slide really just illustrates a handful of those that need to be addressed in order to move the field forward. So for example, whether or not PPAR beta is actually upregulated by this APC beta-catenin pathway is really uncertain. The preponderance of evidence to date suggests that it probably is not as simple as once proposed. One of the most convincing pieces of evidence was provided by Alan Clark's group, who showed that if you delete APC in the intestine, you get an increase in c-Myc and you get an increase in cyclin D1, but you do not see an upregulation of PPAR beta. In fact, he reported a downregulation. So that's an important question that needs to get addressed. Another one is whether or not NSAIDs actually decrease PPAR beta expression. There are papers suggesting that it does. There are also papers suggesting that it doesn't. And that needs to be examined much more critically. Then there is the issue of reconciling how a nuclear receptor that is expressed constitutively at very high levels in tissues such as intestine can function physiologically at one level, but then at higher levels promote tumorigenesis. And that kind of leads into another question that relates to the redundancy in the response elements. Because PPAR gamma, for example, is known to inhibit tumorigenesis, and it's uncertain why activity of one PPAR would inhibit while another one would promote tumorigenesis due to this redundancy. And then lastly, the definitive target genes that either drive tumor promotion or inhibit tumorigenesis have not been identified, and that is another area that needs to be examined much more carefully. So for PPAR beta, it is involved in skeletal muscle, very important to skeletal muscle, fatty acid catabolism. It improves insulin sensitivity, inhibits hepatic lipogenesis. It's improved with weight loss, improving HDL cholesterol. It's an exercise mimetic. It regulates terminal differentiation, and it has anti-inflammatory activities. Its role in cancer is currently uncertain. And that moves on to my last several slides here, and that is to talk about the idea of combining PPAR activities to improve efficacy. Bezafibrate was initially used because it was a PPAR alpha agonist, but it turns out that it actually was the first pan PPAR agonist because it can activate all three of the PPARs, and in fact has greater affinity for PPAR beta as compared to the other two PPARs. This led to the idea that the phenotype observed in the clinic in patients receiving this compound, where you're getting decreases in triglycerides, improvement in HDL cholesterol, improved insulin sensitivity, and no increase in body weight, is likely due to the combined activities of all the different PPARs in causing this effect. And that led to ideas that perhaps you could harness the ability or the activities of different PPARs and improve the efficacy of certain drugs. So one of the first high-affinity dual PPAR agonists described was miraglitazar, or it was part of the glitazar class of drugs, and this was a PPAR gamma, PPAR alpha dual agonist. The efficacy for decreasing hemoglobin A1C was significantly better than that observed for pioglitazone or rosiglitazone, and that was due to activity of PPAR gamma. But the added benefit of this compound, at least they thought, was that it was also able to significantly increase HDL cholesterol, which was something that's a big problem in diabetics. And due to this activity, this drug was thought to be an improvement over the thiazolidinediones. And it gained FDA approval, but shortly after that approval, a report in JAMA came out and showed that the administration of this compound was associated with myocardial infarction, strokes, and congestive heart failure, and the company that made that drug soon thereafter pulled it from the market because they were concerned for that. And they had reason to be. The FDA actually reports that many PPAR agonists that are being submitted to them for trials have actually come up positive in preclinical trials showing the presence of different cancers as well as the adverse side effects that I described earlier. And this has led the FDA to require 2-year carcinogenicity testing for any PPAR agonist on the market. And that's hindered the development of these compounds significantly. So there are a number of challenges facing people out there that are interested in identifying either natural or chemicals that might take advantage of PPAR activity for the improvement of diseases. And this slide here simply illustrates that idea, and that's showing you that here, this is triglydzone here in the blue as compared to rosy and pioglitazone, which are thought to be safe. And you can see that there are some genes that are negatively associated with some phenotypes whose expression is increased by triglydzone, but not so by pioglitazone or rosiglitazone. And this led to the idea that perhaps you can come up with screening tools for coming up with safe gene expression profiles for potential PPAR agonists that might be of benefit and not cause the toxicity observed in the clinic or in preclinical trials. This would need to be coupled, though, with a number of different screening approaches, but this is what's out there. And so I leave you on this last slide, which kind of summarizes, and there's really 3 points I wanted to make from this slide. And the first is that, I hope that you've gained from this presentation, is that PPARs each have distinct and very notable activities that have potential benefit for applying them to different diseases, including metabolic syndrome, diabetes, and dyslipidemias. In case in point, the fibrates and the thiazolidinediones are very effective for treating the dyslipidemias and diabetes that are used in the clinic that activate PPAR alpha or PPAR gamma activity respectively. It remains to be seen whether or not compounds can be identified or developed that take advantage of different activities of PPARs to improve the efficacy of single agents, but that is something that probably needs to be examined. The second point that I'd make from this is that not only combining the beneficial activities of these receptors might be an approach, but also something that might offset. So for example, for PPAR gamma agonists, it's known that they are associated with weight gain and osteoclast activity. So a pure PPAR gamma agonist might be benefit if you develop a PPAR gamma, PPAR beta agonist, due to the fact that PPAR beta agonist activity can offset that by increasing osteoblast activity or increasing lipid catabolism and potentially offset some of these negative effects. So that's another approach that might be considered in the future. And lastly, the third point and final point that I wanted to make is that these types of diseases are going to require very long-term treatments. And because of that, this raises the bar in terms of safety issues. And that's going to be a problem that's going to need to be considered in the near future for any compound that wants to try to take advantage of these based on the evidence in the clinic. And so with that, I will end with my last slide, my acknowledgments. I would just acknowledge the people in my lab who actually get things moving along, in particular Cherie Anderson, who manages the thing for me quite well. The Laboratory of Metabolism here at the NIH, who provided me with my training and the basis for all the work I've ever done. I owe a lot to them in great detail. The NIH for providing me support for my research. Les Biesucker, I'm not sure if he's here or not, but he serendipitously actually made my career-changing mode in my life. He introduced me to Frank, so that was quite a big thing. Gary Purdue, my mentor at Penn State, and last but not least, my family, who provide me the balance in life that's needed to keep things balanced. And that's it. I'll take questions.
Thank you, Jeff. We have time for some questions.
Have you looked at levels of these receptors in macrophages?
Yeah, we're working on that. So do you want to know what they're like, or?
Yeah.
We've never done it quantitatively, but we do know that if you treat macrophages with specific chemicals, you can increase expression, and the one that goes up the most is PPAR beta. But they're all there, and they all have activities in macrophages.
Thank you.
I mean, the kinds of things that are up there, the two that get studied the most are PPAR beta and PPAR gamma.
But I guess specifically, do you see higher peroxide production?
Higher peroxide production due to?
With treatment of the agonists, sorry.
We've never looked at that. No, I would say I would predict the opposite of that.
You see anti-inflammatory.
They're anti-inflammatory,
Okay.
and that's a good point. Activating PPAR gamma or beta in macrophages elicits anti-inflammatory. You get repression of IL-6, TNF alpha, that kind of a thing, with both receptors.
Your summary slide indicates that all the three variants of alpha, beta, and gamma do have beneficial effects, but you also suggested that there are species difference. So this summary of the final slide, is it from animal studies or human studies or both? So this suggests that we should maybe concentrate more on human tissues in some of these evaluations.
Oh, there's no question about that. I think that in terms of what we know about species differences, we could put it in a thimble. We don't. There's not been enough studies done on that. The only PPR that's been studied extensively to dissociate between humans and rodents, for example, is PPAR alpha. And that's because of this cancer issue. There is interest in studying differences between the two of them, but you don't see too much of that being done. I mean, there's lots of studies done in human cell lines, and you see lots of things that compare, but the direct comparisons are rarely made. They do typically, though. I have yet to really see a known difference that in what we study. I've never seen too many marked changes between what we see in rodent cells that we examine and human cell lines that we examine. But there are a couple examples that I can't think of off the top of my head that I've seen that I've thought about that, but it's not been examined much to date.
PPAR alpha aside, relative to the tumorigenicity of these compounds, and just focusing on PPAR gamma and delta, there is a lot, as you reviewed, a lot of data suggesting that they have anti-inflammatory and anti-tumorigenic properties. But could you comment on how we're supposed to reconcile that with the very pro-tumorigenic effect that many of these compounds have in those studies in terms of inducing hemangiosarcomas, liposarcomas, bladder tumors, squamous cell carcinomas?
Yep, that's all coming out. That's all, yeah, that's down the pipeline too. That's an excellent question. I don't think I have a good answer for it either, because I think that in a lot of these 2-year bioassays that are reported, like hemangiosarcoma is a big thorn in the side for many of these compounds, or bladder cancers. The bladder cancer is one where actually is a good example for this potential species difference has been proposed, that the rats have inherent differences in the way their bladder is kind of sitting and why they're going to form crystals. There's been a hypothesis about there to suggest that there might be a difference for the bladder cancer. So that's one mechanism that's been worked out. That's like the only one I can think of that stands out. How do I reconcile why you see papers? I mean, I said that PPAR gamma is chemopreventive, but I mean, I can pull a paper out that was published last year saying the exact opposite, that they found increased tumorigenesis in the colon of animals treated with these agents. And I don't know how to explain that. I mean, other than to suggest that they're either off-target events, serendipitous findings, or there's some problems with them. I mean, I think that if you look at I think part of the problems with the PPRs in general, PPAR beta and gamma or delta as you call it, is that you have to look in the broad scheme of things and look at the body of evidence that's out there developing and keep monitoring it very closely, because the jury's still out there in particular for PPAR delta. PPAR beta is really not, I don't think you can put your hand down and say it's doing this or that at this point. PPAR gamma, though, on the other hand, I think that you can say that it's more of a chemopreventive tool. And I say that with fairly good confidence because there are clinical trials out there that are being tried with people, and they're not finding pro-tumorigenic events in people, or they're stopping those trials. So why you see it in rodents versus the humans probably gets back to the species difference question, and I don't think we have an answer for that, because these receptors were cloned in 1990, and when I first started in Frank's lab, I used to say it's only 5 years ago, and here we are, we're getting to be almost 20 years, and we still don't know much about these receptors. So there's great potential. So I can't answer the question fully. I hope I tried to get it.
There have been recent interest in endocannabinoids, which by and large have opposite effect to PPAR because they, for example, suppress HDL cholesterol and increase insulin resistance. So that raises the question in my mind, is there any example of interaction between, they act through GPCRs, these GPCRs and the nuclear type PPAR receptors at the receptor level? Some kind of an antagonistic interaction?
Yeah, that's a really good question, too. We were interested in that some time ago, and I'm not quite sure. I'm trying to think. I'm scanning my head here. The 2AG, I can't even say the name of the compound. whether or not it does bind to these receptors. And there are some studies that have been reported suggesting that they do. And I was actually trying to think, I don't have a good answer for that question. I think but that idea that the endocannabinoids are functioning through these receptors at either antagonists or agonists is an idea that's out there that's not been studied very, very much to date. And I don't have an answer for it off the top of my head, sorry.
Yeah, Jeff, I have a final question here. The fibrate drugs are very low affinity PPAR alpha activators. Patients take them in hundreds of milligram quantities, and drug companies have been trying to develop, in fact, they have developed very high affinity agonists. And back in the days when I used to be able to talk to drug companies, I heard about several of these compounds that had toxicities that was associated with myotoxicity, that is holes in muscles, particularly of dogs, beagles.
Right.
Do you have any idea about what the mechanism of that could be?
No, there is a paper that just came out trying to describe the mechanism for it. I read it and wasn't very convinced of it all. I don't think anybody knows the answer to that question. Nobody knows the answer to that question, so sorry.
With that, I think we'll adjourn. Thank you, Jeff, for a terrific talk.
Thank you.
And we have a reception back, a couple rooms back here on the first floor. Encourage everyone to attend.